Welcome to Lecture 4. In this lecture, we explore how nature, through centuries of observation and scientific progress, has remained our greatest pharmacy. After this series of lectures, we will learn understand the historical and current role of natural products in drug development, identify key classes and sources of natural products, discuss modern techniques used to isolate, characterize, and optimize natural compounds, appreciate the challenges and future trends in natural product-based drug discovery. First of all, I'm discussing with the natural products and their utility as therapeutics, advantages and disadvantages. Natural products have long served as a cornerstone in the field of medicine. In fact, approximately over half of the pharmaceuticals used in clinical practice today are either derived from or inspired by natural substances. Some of the most significant drugs in modern pharmaceutical care, such as quinine, theophylline, penicillin G, morphine, fluxetazole, digoxin, vincristine, doxorubicin, cyclosporin, and vitamin A, all trace their origins to natural sources. Many of these compounds were discovered because of their traditional medicinal uses, especially from plants that have been used for centuries across cultures to treat a wide range of ailments. This rich legacy of traditional knowledge, combined with modern scientific techniques, has enabled researchers to isolate, validate, and optimize these natural compounds, leading to some of the most effective and life-saving therapies we have today. Starting with the history of drug discovery and development, the early history of medicine. The drug discovery and development has a long and rich history that dates back to the earliest days of human civilization. In ancient times, drugs were more than just physical remedies. They were closely linked to religious rituals and spiritual healing. Often, sages or religious leaders served as healers, administrating plant-based medicines along with minerals and animal-derived substances. Most of these remedies were likely discovered through trial and error, as well as careful observation of human and animal responses. Despite developing independently across civilizations, folk medicines were used similar herbs for treating the same illness. This suggests that ancient traders played a crucial role in spreading medical knowledge during their travels. For centuries, folk medicines remained the only form of treatment available. However, in the late 18th century, the drug discovery began to adopt scientific approaches. Following World War I, the modern pharmaceutical industry emerged, shifting from small-scale natural extraction to large-scale drug synthesis. Yet, even today, many ethnic groups continue to rely on traditional remedies alongside modern pharmaceutical drugs. Traditional Chinese medicine, or TCM, is believed to have originated early 5,000 years ago during the era of legendary Emperor Sino. China's dynasty, culture, and dedication to written records helped preserve ancient medical texts such as Shanghan Long, Wang Di Lei Jing, and Senong Ben Kaojing. The Chinese pharmacopoeia is vast, and some herbal compounds like reserpine from Rafflesia and ephedrine from Mahuang have been incorporated into modern Western medicines for treating hypertension, asthma, and mental health conditions. Egyptian medicine is well documented thanks to ancient papyri. The Ebers Papyrus, dating back to around 3000 BC, contains over 877 prescriptions for various ailments including skin issues, gynecology, and internal disorders. The Count papyrus from around 1800 BC focused specifically on women's health. Egyptians used herbal remedies such as myrrh, frankincense, castor oil, fennel, and garlic, many of which are still recognized for their therapeutic value today. In India, traditional medicine is known as Ayurveda, which dates back 3000 to 5000 years. Rooted in sacred text called the Vedas, Ayurvedic medicine was practiced by Brahmin sages. Its material medica is vast and largely herbal. Some Ayurvedic herbs like cardamom and cinnamon have made their way into Western medicine. A notable historical figure, Sushruta, described the use of henbane as an antivenom for snake bites as early as the 4th century AD. Greek medicine was heavily influenced by earlier systems like those of Egypt, Babylon, India, and China. The Greeks used castor oil as a laxative and linseed for soothing gastrointestinal issues. Fennel relieves intestinal discomfort, and asafoetida serves as an antispasmodic. However, their greatest contribution was the rejection of supernatural explanations for disease. Hippocrates, often called the father of medicine, proposed that illness had natural causes and laid down ethical principles for physicians, principles that still form the basis of medical ethics today. The Romans, known for their organizational skills, built some of the first hospitals primarily for their military. They also adapted and expanded upon Greek pharmaceutical practices. Two prominent Roman physicians, Dioscorides and Galen, were instrumental in advancing medical knowledge. Dioscorides' Materia Medica details remedies composed of 80% plant-based, 10% animal-derived, and 10% mineral-based ingredients. His work remained a cornerstone of Western medicine for centuries. Next, going to the medicine in the Middle Ages and beyond. Between AD 400 and 1500, the Middle Ages marked the decline of Roman influence across Europe. This era was placed quite literally by devastating outbreaks of diseases. Epidemics such as the bubonic plague, leprosy, smallpox, tuberculosis, and scabies swept through communities, claiming the lives of millions. During this turbulent time, the early church played a quiet yet vital role in preserving medical knowledge. While herbal references are scattered throughout the Bible, the church's most important contribution to medicine was the preservation and transcription of ancient Greek medical texts. These preserved works would later become foundational to the medical advancements of the Renaissance. Meanwhile, in the East, the Arabians and Persians were expanding the frontiers of medicine. Trade with various cultures enriched their understanding of diseases and treatments. Perhaps their greatest legacy lies in refining medical preparations and introducing distillation techniques, which may have adapted from early alchemical practices. One of the most influential figures of this era was Avicenna, a Persian polymath who lived around AD 900 to 1000. His medical encyclopedia, the Canon of Medicine, became one of the most authoritative texts in both Eastern and Western medicine for centuries. Another renowned Persian physician, Rhaz, made significant contributions as well. He was the first to clearly distinguish measles from smallpox, marking a turning point in clinical diagnosis. Now come forward to the foundation of current drug discovery and development stages. The Renaissance period marked a turning point in the history of medicine. It laid the foundation for scientific thought and evidence-based practice in both medicinal preparations and treatments. This era witnessed groundbreaking advancement in anatomy, physiology, and surgery, as well as improvements in public health, hygiene, and sanitation, all of which helped shape the future of modern medicine. Smallpox and vaccination. In 1796, a major milestone was achieved by Edward Jenner, who successfully carried out the first smallpox inoculation. His experiment paved the way for the concept of vaccination, offering hope against infectious diseases. Next, the digitalis and cardiac care. Around the same time, William Withering introduced digitalis, an extract from the foxglove plant for treating heart conditions. It marked one of the earliest uses of a plant-based drug in modern cardiology. Scurvy and nutritional deficiency. In 1753, naval physician James Lind discovered that scurvy was caused by a lack of vitamin C. His recommendations to use lemon juice for treatment highlighted the importance of diet in disease prevention. At the rest of the rabies and microbial theory, the latter in 1864, Louis Pasteur proved that microorganisms were the cause of many diseases. He developed a vaccine for rabies using an attenuated virus, setting the stage for modern microbiology and immunology. Revolution of drug discovery and the rise of modern pharmaceutical. Despite scientific advancements in the 18th centuries, the range of drugs available in the early 19th centuries was still very limited. Some of the commonly used drugs included digitalis from the foxglove plant used to stimulate cardiac muscles and treat heart conditions. Quinine, extracted from the cinchona tree, was used to treat malaria. Ipecacuanha, obtained from the syphilis plant, treated dysentery. Aspirin, derived from the willow bark, provided relief for fever and inflammation. And mercury, despite its toxicity, was used for treating syphilis. The systematic drug research begins. In the early 19th centuries, drug development began to follow a more scientific and systematic approach. Paul Ehrlich introduced asphenamine and arsenic-based compounds to treat syphilis. Later, Gerhard Domag discovered that prontosil, a red dye, could kill streptococcal bacteria. French scientists then isolated its active component, sulfanilamide, which led to the development of sulfur drugs, a whole new class of antibacterial agents. After that, the discovery of penicillin. In 1928, Alexander Fleming discovered the antibacterial effect of penicillium mold on Staphylococcus bacteria. A decade later, this was re-examined by Ernst Chain and Howard Florey, who together made possible the large-scale production of penicillin by 1944. This milestone marked the beginning of biotechnology, where microorganisms were harnessed to produce drug products. The shift from herbs to synthetics and biotech. Until the late 18th centuries, most drugs were made from herbs and natural extracts. But in the early 19th centuries, the pharmaceutical industry began focusing on chemical synthesis of medicines. These drugs were typically therapeutic, not curative. From the 1930s onwards, the drug discovery focused on screening natural sources for active ingredients and then synthesizing them into new chemical entities, or NCEs. These synthetic versions had to undergo rigorous testing for safety, potency, and efficacy. Entering the era of biotechnology, by the late 1970s, scientists began developing recombinant DNA products utilizing molecular biology techniques. This gave birth to the biotechnology industry. Today, the pharmaceutical landscape continues to evolve, driven by gene therapy, targeted drug delivery, and insights from the Human Genome Project. These innovations enable the creation of drugs that can specifically target disease sites, making a new era of precision medicine. This progression from ancient herbs to synthetic molecules and now to biotech represents a remarkable journey in our fight against diseases and in the advancement of human health. Nature continues to guide us from ancient herbs to modern labs. The journey of drug discovery is far from over. The natural products remain its heart. Thank you for joining. Up next, we will explore the modern techniques used to extract and modify these natural treasures and the how to develop or discover natural leads. Stay tuned after the next